Okay, welcome to the video for chapter 6.1. So chapter 6 is pretty important, and especially section 6.1, because we're going to be learning a lot about what we do in the lab. So there's a lot of direct correlation to the lab part of the course in this first section. Okay, so section 6.1 is all about culturing and counting bacteria. You might want to get a calculator. We are going to do some calculations in this lecture. Okay, so chapter 6 objectives, we're going to learn all about bacterial growth, nutrition, and differentiation. Okay, so we're going to be able to explain how nutrition and the environment can impact microbial growth and differentiation. We're going to discuss microbial classifications based on nutritional needs and environmental limits. And we're going to describe how understanding microbial growth helps identify disease-causing pathogens. And then finally, we're going to discuss biofilms and their importance to infectious disease and why they're such a problem in the medical community right now. Okay, so let's talk about this chapter's case study. This case study is called metabolic fingerprints of a brain pathogen. Okay, so let's set the scene. So the scene's going to start in a New York City emergency room. So a young mother who recently immigrated from Uganda has brought her 18-month-old daughter, Mirembe, to this ER room. Now, the infant is constantly crying uncontrollably, and obviously the infant's in pain. Okay, so unfortunately, the thing about infants is you can't say where does it hurt and have them tell you where it hurts. So what the on-call doctor is going to do is they're going to start to move Mirembe's body around and see where the crying gets worse, because that's going to be an indicator of where she's hurting. Okay, so the doctor starts moving Mirembe around, and he goes to turn her head, and she starts really crying. Okay, so between the pain in the neck and a couple other things that they look at, the doctor is starting to suspect that Mirembe has meningitis. Okay, so this is an infection of the cerebral spinal fluid and the meningeal layer around the brain. Okay, so this is what he's kind of thinking. So we need to run a few tests to back up his hypothesis that this is meningitis. Okay, so we're going to do a bunch of tests. So the first thing that's going to happen is that they're going to collect a spinal tap. So they're going to insert a needle in Mirembe's back, and they're going to pull out some of the cerebral spinal fluid, the CSF. Okay, so if you guys remember, it's been a few chapters, but we saw an example of normal versus infected cerebral spinal fluid. So in the vials, one was clear and one was cloudy. Okay, so if she really does have an infection that is affecting her meningeal layer, if it's really meningitis, we expect to see a clouded CSF sample. Okay, this is not going to be enough because we really need to know what it is so we can prescribe antibiotics for Mirembe. Okay, so we're going to collect a sample and we're going to gram stain it. Okay, so we're going to look at it underneath the microscope. Okay, so the microscope's going to reveal that we have a gram-negative bacilli. Okay, so it's going to be rod-shaped. Now, there's a lot of gram-negative bacilli that are pathogens, like we have E. coli and a bunch of others. Okay, so we want a more definitive answer than this, but we're getting closer. So based on the fact that this was a gram-negative bacilli, the hospital is going to choose a bunch of solid media. So our petri dishes here. Okay, this is what we mean by solid media. It's agar in a petri dish. Okay, so they're going to choose a selection of petri dishes, and they're going to culture this sample on a bunch of different media. Okay, so we have these two media here. This one on top is called a blood agar, and this one's called chocolate agar. Okay, so both of these have sheep's blood in them. So this top one has intact whole red blood cells. Okay, because there are some bacteria that will break open the red blood cells to get to the iron, and that's one way we can tell what type of bacteria we have. And with the chocolate agar, it's called chocolate because it looks like chocolate. It does not have chocolate in it. What it actually contains is red blood cells that we have lysed open. Okay, so it's the iron's easily accessible. The bacterial cell doesn't have to break it open itself. Okay, so what they found when they did these tests on the solid media is that nothing grew on the blood agar with the sheep's blood. Okay, but they did get bacterial growth on the chocolate agar and a different agar. So there was a a third agar that is going to contain specific growth factors, known as X and V. We were very, very imaginative when we name things in microbiology. So we get growth on that media with the growth factors on it. Okay, so between the no growth on the blood agar and the growth on the chocolate agar and the media with the X and V in it, the doctors can make a diagnosis. So based on the metabolic pattern, what they see underneath the microscope, what they see on the plates, okay, the diagnosis is that in fact Mirembe has meningitis, and her meningitis is caused by a strain of bacteria called Haemophilus influenza. Okay, and once they have this diagnosis, they can give specific antibiotics to Mirembe, and they did this, and she makes a full recovery. Now, we need to talk a little bit more about Haemophilus influenza, especially Haemophilus influenza B, was sometimes referred to as HIV, not HIV, HIV. Okay, so this is a very invasive strain of Haemophilus, and it's going to be directly transmitted by direct contact or via the airborne route. So prior to 1988, when a vaccine was developed, HIV was one of the major causes of childhood meningitis throughout the whole world, third world countries and developed countries like ours. Okay, unfortunately for Mirembe, even though this vaccine has been out since 1988, all of you have probably gotten it. I got it when I was a kid. Mirembe, on the other hand, was from Uganda. They did not have access to this vaccination. So when she moved to America, she was susceptible to getting this infection because she was not vaccinated for it. Okay, so let's go into 6.1. This is all about culturing and counting bacteria. This is a very lab heavy section. We're going to learn about a lot of stuff that we are going to be doing in the lab. In fact, lab 5 is a lot of what we see here. Okay, and that's going to be our Friday lab because it does take some time. Okay, so the objectives for section 6.1 is we need to be able to explain how pure cultures are obtained and why they are important in medicine. We need to be able to distinguish among synthetic, complex, selective, and differential media, and their different uses in clinical microbiology. There's a lot of vocabulary in this chapter, guys. And there's some math. And we're going to describe the ways bacterial growth is measured and explain the advantages versus disadvantages of each method. Okay, so let's talk about how we grow bacteria in culture. So a lot of times when we collect a sample, especially from nature or from a medical sample like a stool sample, there's a lot of different species of bacteria in those samples. In fact, in nature, microbes are going to exist in very complex, large multi-species communities. So there could be up to hundreds of different bacteria in a sample. Okay, to study something in a lab or to find out what's making something sick or somebody sick, we are going to have to be able to separate the different species from each other. So let's say that we collected a stool sample, so a sample of poop from a patient, and there's all these different types of bacteria in there. What we're going to do is we're going to use specific types of culture. So culture is how we grow bacteria in the lab, and we are going to specifically use a culture that's going to allow us to separate the different bacterial species from each other. Okay, so if we look here at our little plate, okay, we have a bunch of different types of bacteria here. We have this yellow guy, we have this like pink guy, we have this salmon guy, we have this orange guy, we have the little white and cream guys. Okay, So this would be an example of a plated mixture. Okay, so when we're doing a laboratory study, we want to have a pure culture. So we want to grow something that only has a single species in it so we can study it, or we want to be able to isolate a single thing to study it. Now, all these little dots here, these single dots, are what we call a colony. Okay, each colony is going to start as a single cell that is going to reproduce clonally, meaning it's going to grow and divide by binary fission. And this little colony is going to be made up of all those bacterial cells that have divided from that initial single cell. Okay, so these colonies are going to be a single species, and they're going to be genetically identical because that's what happens when you go through binary fission. Okay, so if you remember back to Koch's postulates a few chapters ago, one of the steps was to get a pure culture so that we knew that the pathogen so that we could isolate the pathogen that was really causing disease. Okay, so we do the same thing in the lab. Okay, so if we take our inoculating loop and we scoop this guy out and we put him into a pure culture, into a culture but all by himself, we will get a pure culture. We'll get something that's only this bacteria here. We could do the same with this guy, with this guy, with this guy. As long as they're not crowding together, kind of like these guys here, we can use them to build a pure culture. Now, in the lab, we have different culture media. Okay, we can grow bacteria in either a liquid culture or a solid culture. So each type of culture has its own pros and cons. Okay, so with liquid media, this is really great. It allows bacteria to move freely, they grow pretty quickly. Unfortunately for liquid media, it's very hard to separate a mixture in liquid media. So to do that, we're actually going to use solid media like our agar plates, our petri dishes, because we can use the petri dish to separate things out. So we can start to get individual colonies so we can get that pure culture from those individual colonies. In fact, we are going to be doing this in the lab. You are going to be given a mixture and you're going to have to use a agar plate to get a pure culture. Or sorry, you're going to have to separate things out from each other and so you can make a pure culture. Okay, so let's talk about how we obtain pure cultures. So a lot of times we're going to have a mixture and it's going to be in the liquid media. So this is going to be something like broth. Okay, so it's the easiest way to get enough organisms to plate. Okay, so we want the bacterial cells to form colonies on the solid media. Okay, that's going to give us a solid surface that we can easily isolate them from each other. So when we are trying to separate a mixture of bacterial species, we're going to use a technique called isolation streaking. We are going to do this in the lab. It's pretty fun. Okay, so isolation streaking is going to allow us to separate a mixture into individual pure colonies. Okay, and just a reminder, a colony is going to start out as a single cell, and it's going to clonally divide by binary. So go through clonal reproduction or binary fission, and it's going to grow and reproduce and create identical copies of itself. So we know that each colony is pure. It's going to only contain one species. Okay, we also have this term in microbiology because sometimes we don't have a single cell that starts a colony. So we have things like staphylococci and other things that grow in chains or clusters that they're never going to be just one single cell making the colony. So we actually use the term CFU or colony forming unit to indicate that it's going to be a pure culture. It just didn't start with a single bacterial cell. Okay, so let's talk about how we do isolation streaking. So what we're going to do is we are going to have our sample in the liquid media in our broth sample. Okay, we are going to turn the Bunsen burner on. We're going to have our petri dish with no growth on it, and we're going to get our inoculating loop. Okay, so we're going to use aseptic technique. We're going to flame the outside of the liquid media. We are going to do the cap, flame that again. We're going to make sure our inoculating loop is flamed, so it's nice and sterile. We're going to use it to collect a loop full of the sample from the broth. And what we're going to do is we're going to do the first streak. Okay, so we're going to do this part. So we're going to go up and down until we have transferred the culture from the container to the dish. Okay, so we're going to close our clamshell, keep things aseptic. We're going to flame our loop again, and this is important. We're going to flame it, and we're not going to get any more sample. No more sample. We only dip it for the first streak. Okay, so once we have our inoculating loop flamed again, what we're going to do is we're going to rotate our petri dish by a quarter, and we're going to do this streak again. Okay, so we're going to take our clean loop and we're going to drag it through the previous sample that we applied to the agar. So what this is going to do is we're going to go from something where the colonies are really close together and we can't see individual ones, and they're going to start to separate them out. Now, we're going to do this 3 or 4 times. We're going to work our way around the plate. Okay, so we're going to do this streak. Okay, we're going to close our clamshell. We're going to flame our loop again. We're going to turn the petri dish another quarter turn. We're going to take our now sterile inoculating loop and we're going to drag it through this part of the streak. So we're going to have a third streak. So now what we're seeing is we're starting to see individual colonies. So a lot of times we're going to flame our loop again and turn it one more time. Okay, and we're going to get this separation here in the center. Okay, so this is how we get individual colonies to make pure culture from a culture that we know that is a mixture. Okay, but the important thing to remember about this, and you will be tested on this in both the lab and in the class, is that you do not get more sample after that first transfer. Okay, we're going to keep using the loop and keep streaking the plate, but we are not going to add more sample to it. Okay, that would defeat the purpose of trying to separate it out into colonies. Okay, and we need to reflame every time. Okay, so we make sure it's sterile so we're not just dragging a bunch of bacteria around the cell, because if we don't flame it, we're not going to get this separation. So we're going to watch a animation about isolation streaking. Sometimes this is also called dilution streaking. We're going to call it isolation streaking.
Agar medium in a petri dish provides a large flat surface on which a mixture of microorganisms can be streaked to separate individual cells. In the dilution streaking technique, an inoculating loop is sterilized in a flame and allowed to cool. The loop is used to collect a drop of liquid from a bacterial culture. With the loop held flat on the agar, the drop of liquid is streaked across a region of the agar plate's surface. As the loop moves, organisms begin to rub off the loop. The loop is sterilized and cooled again. This time, the loop streaks over the first pass, collecting a small number of bacteria and spreading them into a fresh area of the dish. The procedure is repeated again. Toward the end of the streak, few bacteria remain on the loop, so at that point, individual cells will land and stick to different places on the agar surface. If the medium contains the proper nutrients and growth factors, a single cell will multiply into many millions of offspring, forming a colony. In general, a pure culture can now be obtained by touching a single colony with a sterile loop and inserting that loop into fresh liquid medium. It is important to note that the one cell equals one colony paradigm does not hold for all bacteria. Organisms such as streptococci or staphylococci usually occur as chains or clusters of several cells. A cluster of 10 or so cells, called a colony-forming unit, or CFU, may give rise to a single colony.
Okay, I really like that animation. It shows you what exactly we mean by the isolation streaking technique. So that is how we can take a mixture and get a pure culture from it and isolate it from everything else. That is something we're going to be doing in the lab. So if that's how we get a pure culture, how do we count cells? How do we know how many cells are in a culture? Okay, so how much bacteria is there? So let's talk about this, because the way that we determine how many bacteria are in a sample is we use what is called a dilution. Now, if you've ever gone to the freezer section of the grocery store, or if your mom has, they have they may have bought a can of frozen orange juice concentrate. Now, if you eat frozen orange juice concentrate straight out of the can, it doesn't taste very great. It's very sour and very cold. So a lot of times what we're going to do is we're going to follow the directions on the can, and we're going to take the frozen concentrate and mix it with water and dilute it down into something that more resembles orange juice as we know it. And that's good and tasty and way better than just eating it straight out of the can. Okay, we're going to do something similar here with bacteria to determine how many are in a sample. Now, I'm going to walk you through the math, so you might want to get your calculator out. Okay, so we are going to start with 10 milliliters of a culture that has 10 bacteria in it. Okay, so this is my culture. Got my 1,000 bacteria in here. Okay, so we're going to start with 10 milliliters of this culture in this flask. Okay, now we want to know how many bacteria are going to be in 1 milliliter. So what I want you to do, I want you to pause the video, and I want you to see if you can determine how many bacteria are in a milliliter. Let's see if you can do the math. Okay. Well, the math for this is pretty easy. We chose some pretty easy numbers. So if we have a sample that has 1,000 bacteria in it, okay, and we take 10 milliliters of that sample. So if we want to figure out how many bacteria per milliliter, all we have to do is we take this 1,000 and we divide it by the 10 milliliters. Okay, so when we do that, a 0 is going to cancel, and we are left with 100 bacteria in that sample. Now, we're going to say we statistically have 100 bacteria in that sample. It could be plus or minus a few. The real world's not as nice as the math we're doing here. Obviously, we're using tens. Tens are easy math. Okay, so we have 100 bacteria per milliliter in our concentrated sample. This is not dilute yet. This is not the dilution. This is where we're starting. Okay, so we know that in our concentrated sample, we have 100 bacteria for every 1 milliliter of that sample to get us to that 1,000. Okay, so what if we take 1 milliliter of our concentrated sample? Okay, so that's going to be 1 milliliter with 100 bacteria in it. We take it and we put it in its own clean test tube. Okay, so we take 1 ml, and then to this 1 ml, we're going to add 9 milliliters of fresh liquid broth. Okay, so this is going to be a total of 10 milliliters of broth. Okay, so we took this 100 bacteria and we diluted it down into 10 milliliters. Okay, this is what we call a 1 to 10 dilution. So we sometimes see dilutions like this, like a ratio. Sometimes you see it as a fraction, so 1 divided by 10. It's all the same thing. Now, this can get to be a little confusing because sometimes you want to think, oh, 1 plus 10, we have 11. No. No, 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 no. Okay, so when we have a 1 to 10 dilution, I want you to start thinking about it like this. We have 1 milliliter that became 10 milliliters. Okay, so 1 milliliter of culture plus 9 milliliters of fresh broth gives us 10 total milliliters. So 1 to 10 means 1 milliliter becomes 10 milliliters. Okay, so we're going to add the math to the slide. So we have our concentrated solution here. So this is our 1,000 bacteria. This is our first 1 to 10 dilution. Okay, so we have 100 bacteria in this 10 milliliter sample. Now, what happens if we take 1 milliliter of our 1 to 10 dilution? Now, sometimes, as I said, you see it as 1:10. Sometimes you see it written as a fraction. Sometimes you see the exponent form. Okay, so this is our 1 to 10 dilution. So what if we take 1 milliliter out of this 10 milliliters? What is our concentration of bacteria per milliliter now? Pause the video and see if you can figure out the math. I'll give you a hint. It's going to be very similar to the math we did previously. Hey, do you have an answer? Do you know how many bacteria are in that 1 milliliter of the 1 to 10 dilution? Well, let's do the math. Okay, we have a 1 to 10 dilution that had 100 bacteria in it. We took 1 milliliter out, so we're going to do the same math again. So we have 100 divided by 10 milliliters. So 100 divided by 10 is going to be 10 bacteria per milliliter statistically. Remember, we can have plus or minus a few in the real life. Okay, so we have 10 bacteria per milliliter in our 1 to 10 dilution. So what if we take that 1 milliliter that 1 milliliter from our 1 to 10 dilution, we put it into a new test tube. So we have 1 ml of culture, and then we add 9 milliliters of fresh to it. Okay, what is the concentration in this test tube now? Okay, so we took a 1 to 10 dilution, and we made a new 1 to 10 dilution. So we now have made a 1 to 100 dilution. Okay, so what is the concentration? Well, it's the same, right? We now have 10 bacteria in that 10 milliliters. Okay, so we now have a 1 to 100 dilution. Okay, so we have our 1 to 100 dilution here, the second one. Okay, it has 10 total bacteria in it. What if we take 1 milliliter from our 1 to 100 dilution, the second one, and we take 1 milliliter from that and put it into a new test tube, and we do another 1 to 10 dilution. So we're now going to have a 1 to 1,000th. You're starting to see the math. 1/10 times 1/10 times 1/10 equals 1/1000th, or 10 to the minus third. So what would be the concentration of bacteria per milliliter in our new 1 to 10 dilution, our 1 to 1,000 dilution? Okay, pause the video, do the math. Okay, well, we had 10 bacteria in our 1 to 100. We took 1 milliliter out, we diluted it to 10, we made it 10 milliliters, so we have 10 divided by 10. So statistically, on our 1/1000th, okay, our 1 to 1,000 dilution, we statistically should have about 1 bacteria per milliliter in our concentration. This is what we call dilution. So we went from super concentrated, so we went from 100 bacteria cells in the concentrated mixture. We diluted it down to 10, and then we diluted it down to 1. This is what a dilution does. Okay, we are going to do these in the lab. Now, real world, we are not going to know the concentration, how many bacteria we have in our starting liquid media. Okay, so we are going to use what is called the spread plate technique to figure out how many bacteria we have in our unknown concentration of a sample. Okay, we're going to do these tenfold dilutions. So 1 milliliter from the concentrated into 9 milliliters of fresh broth over and over and over again. Okay, but what we're going to do is when we do these dilutions is for every dilution, we are going to put it on an agar plate. This is why it's called a spread plate. So what we're going to do is we'll take 1 milliliter after we've done the dilution. So we're going to have 10. So we're going to take 1 milliliter from here, and we're going to plate it on the agar plate, and we're going to grow it and see how many colonies we get. Now, our first dilution is going to be still pretty concentrated, right? When we have our early dilutions, they're going to show what we call confluent growth, meaning we're going to have a lot of bacteria on that plate. In fact, we have so much, we call it TNTC, too numerous to count. We're going to grow what's called a lawn. we aren't going to be able to distinguish individual colonies from each other. We're not going to be able to count it. Okay, so what we're going to do is we're going to plate a little bit of our media from each of these dilutions. Okay, so maybe we went from our lawn to a petri dish that has 254 colonies. Okay, and then we do the dilution again, and the next one has 23 colonies in it. Okay, and we do the dilution again, and maybe we get 3. Are you starting to see the pattern in the math here? Okay, so if we use tenfold dilutions, we are going to get somewhere around a tenth less colonies on the plate. So 254 should be somewhere around 25 if we divide it by 10. So we got 23. So not quite decreasing by a factor of 10 there. And then we go from 23 to 3. It should be somewhere around 2 if math worked out perfectly. It doesn't. That's okay. We're close enough to use it. Okay, so when we get down to where we have easily counted colonies, we've got to remember these colonies on these agar plates are going to represent one viable organism. What does it mean when I say viable? Viable means that the cell is capable of reproducing, so going through cell division, doing that clonal reproduction to give living cells to make a colony. Now, when we're using the spread plate technique to quantify how many bacteria we have in an unknown sample, this is only counting the viable cells. So the ones that can grow, the ones that are still living. This does not count dead cells. Okay, we are going to use a different method to count dead cells. Okay, so this pattern of diluting our sample by 10 makes it so it's pretty easy to use the math. Okay, because what we can do is we can take the number of colonies on the plate and our dilution factor. Okay, so what dilution this is, right? This is 110, 1 to 100, 1 to 1000, 1 to 10,000, 1 to 100,000, and 1 to 1 million. Okay, we can take these and we can use the math to figure out how many cells, how many CFUs, how many colony forming units we have in the original unknown sample. Okay, now let's be real. Rarely are dilutions going to give us perfect numbers, but we can get a close estimate of it, okay, by working backwards. So how do we do this? How do we do this math? Okay, so here's a real world example, and this is going to kind of illustrate what it means to have a lawn. Okay, so we did our tenfold serial dilution. Okay, this is our 1 to 10, this is our 1 to 100, our 1 to 1000, our 1 to 10, oh, where's my mouse? 1 to 10,000, 1 to 100,000, and this is going to be our 1 to 1 million. Hopefully I did my math correct. Okay, so if we look at here at our first dilution, so we're going to plate 1 milliliter of each of these dilutions onto the agar plate. Now, this is called the spread plate technique. I probably should have mentioned this earlier. It's because we're going to take a device called a spread bar, and we're literally going to take this 1 milliliter and we are going to spread it all over the plate so we can try to get some separation so we can get individual colonies to count. Okay, so these first two are what we call TNTC, too numerous to count. This is our lawn. We still have a lawn. But you can see with each dilution, we're starting to see more and more colonies being on their own. But this is still way too numerous to count. This is still too numerous to count. We could count this one, this 10 to the minus fourth one. It would take us a long time. We don't have time for that. So we're going to do another dilution. This one's this one will take us a minute. So what we're going to do is we're going to take our marker or our pen and we're going to put a dot on each colony and we're going to count the dots. Okay, so some labs use a clicker. So every time you count one, you click it and it's going to keep count for you. We're not that fancy in our lab. We are just going to count. Okay, so we have this one, and in this example, somebody else counted for us. So this plate has 107 colonies on it. Okay, in the next dilution, in the next tenfold dilution, we are going to have 11. So it's not quite a decrease by 10, but it's close. Okay, so statistic-wise, we are not going to use this one, even though it's easier to count. We want to use about 50 to about 330 count-wise. So we're going to use this dilution here, this 10 to the minus fifth dilution, to figure out how concentrated our sample was. So how many bacteria were in that sample. Okay, so what we're going to do is we're going to take this 107. This was the 10 to the minus fifth dilution. This is our dilution factor. We're going to take the number of colonies times our dilution factor. And that's it. That tells us how many. So we're going to get 107 times 10 to the fifth, because remember, this is the dilution. To go back to concentrated, we have to flip it. Okay, so instead of 10 to the minus fifth, it's going to be 10 to the fifth. So 107 times 10 to the fifth is going to give us 1.07 times 10 to the seventh CFUs per milliliter. Okay, so if we write it not in scientific notation, that is 10,700,000 cells per milliliter. Okay, that's quite a lot. We are going to do this in the lab. Okay, we've had some years where it's worked pretty good. Last year we did really well. We were able to figure out how many cells were in the sample. So this is something we're going to do. This is lab 5. Okay, doing the spread plate technique, doing an isolation streak. That's all part of lab 5. Okay, so if 1 milliliter was plated, that means on each, so our 107 times 10 to the fifth is that 1.07 times 10 to the seventh CFUs. Okay, remember we're using this CFU unit, this colony forming unit, because bacteria don't always move as single individual cells. Sometimes they do, sometimes they don't. So sometimes they move as groups. So we have like the staphylococcus cells. Okay, so we have like 2 here in this bunch, we have 3 here, we have quite a few here, at least 10 here. Okay, but they're all going to be the same thing. So we can't guarantee that colony. So our one cell, one colony paradigm doesn't exactly work. We can guarantee that we have one colony forming unit, and that's what each bunch is. So these 2 are one colony forming unit. These 4 are one colony forming unit. These 3 are one CFU. Okay, they're not going to be one cell starting a colony, but it's going to be like cells starting a colony or one colony forming unit. Okay, so Streptococcus, Staphylococcus, they're all going to have multiple bacteria cells starting a colony. Okay, so here we have 10,700,000 colony forming units in our unknown culture. Okay, so we've talked about how we could get a pure culture. We've talked about isolation streaking. We've talked about the spread plate technique. Now we need to talk about other ways we use media or culture to figure out what we're growing. Okay, so we have different synthetic and complex medias, and we need to know what this means. So if we use a synthetic medium, bacteria are going to be grown on a petri dish that has been made of an agar that we know exactly what that agar contains. Okay, it's only known components. We know how much salt, we know how much gelatin, we know everything that's in it. Okay, so it's precisely defined. This is what we mean by a synthetic medium. Okay, we can have what is called a minimal medium. This is a form of a synthetic medium. Okay, so instead of knowing exactly what everything is and precisely defining it, a minimal media is going to limit the media, so what's in it, to only the things that are going to allow an organism to grow. So water, salt, a carbon source, so like glucose, a nitrogen source, and an energy source. Okay, so it's going to take a synthetic medium and it's going to minimize it. It's going to only include what the cell needs to grow. Okay, bacteria can be grown on non-synthetic media. So this is what we call complex media, okay, or a rich medium. Okay, it's very nutrient rich, but it's less defined. We really don't know what's in it. We just know it grows things. Okay, so with complex media, this could literally have beef extract in it or yeast extract to add those nitrogen and carbon sources and those energy sources. So with complex media, we don't know everything that's in it. With synthetic media, we do. Okay, now we have an enriched media. Okay, these are important. So bacteria can be fastidious. Okay, another term for fastidious, you need to know this term by the way, is fussy. So we can have some pretty fussy bacteria, which means that they have extra components that we need to add to our basic media in order for them to grow. Okay, so different bacteria have different nutritional requirements, and some of them, these fastidious, these fussy ones, we have to add those extra things to the media or they won't grow. Okay, I would know these four definitions for sure. This will show up in the lab, this will show up on the test. Now, that's synthetic versus complex media. Let's talk about selective and differential media. We've kind of talked about this a little bit, but let's go into a little bit more detail. So in lab 3 and in this lecture, we're going to talk about some selective and differential media and what that means. And we're going to look at real world examples. Okay, so just a reminder, selective media, this is going to have compounds in the media that is going to prevent some bacteria from growing while allowing other types to grow. Okay, so they're going to favor the growth of one specific type. Okay, so easy way to remember it, selective media lets some things grow, but not others. Okay, this is why it's selective. It selects a certain type of bacteria to grow. Then we have differential media. Okay, so differential media is going to grow a species equally well, but what's going to happen is we're going to add stuff to the media that is going to allow color changes so we can see how different bacteria metabolize things differently. Okay, we are going to look for the different colors. Okay, so it's differential because it differentiates between two types by a color change. We love color indicators in biology and in chemistry. They make our lives easier. Okay, so this is going to bring us to a specific example. This is called McConkey media. Okay, so McConkey media is both selective and differential. We can have media that's just selective, and we can have media that's just differential, but a lot of times we're going to talk about both selective and differential media. Okay, so in McConkey media, it's going to prevent gram positives and other bacteria from growing, and it's going to allow gram-negative enteric bacteria, meaning gram-negatives that are found in your gut, to grow. Okay, so what it has in it that makes it selective are bile salts and crystal violet. So the same thing we use for the gram stain is going to be in this media. Okay, that's what's going to make it selective. The crystal violet and the bile salts are going to, the bile salts more than the crystal violet, are going to prevent the gram positive cells from growing while allowing the gram negative cells to grow. Okay, McConkey media is differential. Okay, so how can we tell the difference between all these gram negative enteric bacteria? Okay, we are going to add lactose and a indicator because some bacteria are going to ferment lactose while others are not. Okay, so if a bacteria cell ferments lactose, it's going to be pink, okay, because of color changes in the media. If it does not ferment lactose, it's going to show up as a clear or a white colony on the agar. Okay, so we need to be careful because selection is a common term used. So let's look at this a little bit more in depth. Let's look more in depth, not in depth. Let's look at a case study about when this media would be used. Okay, so we have a 15-year-old that goes to the ER. They come in complaining about severe abdominal cramps and they've had diarrhea for 2 days. So the first thing that the ER nurses and doctor are going to do, they're going to collect a stool sample, a fecal sample. Okay, so they're going to take the patient's poop and they're going to streak it on a McConkey plate. Okay, so remember, these McConkey plates only select for gram negatives. Okay, gram negatives are usually a common pathogen for gastric issues, which is why they're going to immediately streak the McConkey plate. Okay, so we're only going to get enteric, so gut gram negatives that grow. Now, our guts normally have gram negatives in our guts that we rely on. We have a symbiotic relationship with those gram negatives. The thing is, we can tell a difference between the gram negatives that are in our intestinal tract versus the pathogenic variety because we've been drinking milk since birth. Okay, milk has lactose in it. So our common gut bacteria, so like E. coli, is going to break down this lactose, okay, because it's adapted to using that as an energy source. Okay, pathogenic varieties are not going to be able to break lactose down. Okay, so they are non-fermenting. Okay, so the ones that are supposed to be there will ferment. Those ones that don't shouldn't be there. Okay, so what we're going to see is we're going to have these bread colonies. These are going to be E. coli gram negatives. They should be there. They are breaking down the lactose. They're fermenting it. That means they can use it. That means that they are part of our normal gut biome. Now, these white ones are going to be Salmonella enterica. Okay, so this is a common contaminant in like leafy vegetables. Okay, some Salmonella contamination is a common recall for those because it's really hard to get them washed off and out of all the nooks and crannies of lettuce. Okay, the Salmonella enterica is pathogenic. It has not been in our guts. It is the invader. Okay, so it can't break down lactose. So this is going to differentiate between our normal gram negatives in our gut and the pathogenic variety. Okay, let's talk about a last case history for 6.1. Okay, so this is called urine cultures when numbers count. So we have Melissa. She's a 25-year-old who has made a doctor's appointment and is visiting her doctor because she has this terrible burning sensation when she goes to the bathroom when she urinates. Okay, so this is a pretty common symptom. So the PA that is seeing her at her doctor's office suspects that Melissa has a UTI or a urinary tract infection. Okay, so they do a clean catch. They have Melissa pee in a cup and they take a sample of her urine to the laboratory. So one of the things that the lab tech is going to do is they are going to take a very small but exact volume of urine. So probably about 100 microliters, so very small amount. And they're going to use the spread plate technique and they're going to spread the urine on the agar medium and they're going to incubate it for 24 hours. So they're going to let it grow. So when they do this with Melissa's sample, the clinical microbiologist in the lab found that Melissa has large numbers of E. coli colonies. So E. coli is a gram-negative bacillus. It's a rod-shaped gram-negative. So he found approximately 100,000 bacteria per milliliter in Melissa's urine. This is a pretty high number. This is not good for Melissa. This is bad. Okay, especially because urine is one of our sterile sites. So we've talked about cerebral spinal fluid quite a bit and how there should be no bacterial growth in it normally. It should be clear. Your urine is also a sterile site like CSF. So there should be little to no bacteria in your urine. Okay, so when you have bacteria in your urine, that is going to be signs of a infection like Melissa's UTI. Okay, because she has so many E. coli in her urine sample, the PA is going to diagnose her with a UTI, and he's going to prescribe an antibiotic that's specific to these uropathogenic E. coli, the E. coli that's causing the UTI. Now, this case history shows how we can use a technique we have in lab and apply it to a clinical setting. Okay, it is very important that we know how concentrated a urine sample is in a patient because it's going to dictate what kind of antibiotics we give the patient. Now, 100,000 is quite a large number, but Melissa can still take oral antibiotics, so she can take the pill form of antibiotics. Now, if her urine sample showed a higher number of bacteria in per milliliter, what was going to happen is if the bacterial count is high, she's not going to be able to take a pill antibiotic. She's going to need intravenous or IV antibiotics that are a lot stronger and that will work more quickly to counteract that infection, because that is a huge infection if you have more than 100,000. Okay, because the problem is if these UTIs are allowed to continue, Melissa could go septic, so she could have a full body infection and it will cause her organs and things to start shutting down. Okay, so these are the indirect ways of counting. So using our plate technique, we can get a pretty good estimate of the concentration of a sample. Downside is it only tells us how many living cells, not how many dead cells we have in a sample. Okay, and it takes some time. We have to have growth. We have to have time for it to incubate. Okay, so the spread plate technique or dilution spread plating is going to rely on growth. Okay, and we're going to have to make those dilutions to figure out how many, and then we're going to have to do the math. Okay, it does give us a pretty accurate number if done correctly. So if we got 100 colonies from 100 microliters, okay, that's going to be roughly 1 cell per microliter. Okay, so if we have 1 milliliter, 1 milliliter is 1,000 microliters, so that would be 1,000 cells per milliliter. Okay, so if this was already diluted by a 1 to 1,000 dilution, then those 1,000 cells are going to be multiplied by 10 to the third, because 1/1000th. So that's going to give us that 100,000 cells per milliliter that the clinical microbiologist was seeing in Melissa's case. Okay, so indirect ways of counting only count viable, and they rely on growth. They take time. Now, what if we need to make a diagnosis more quickly? What are we going to use? We actually are going to use something called a spectrophotometer. And a spectrophotometer is going to measure what we call optical density. Optical density is just how cloudy something is. Now, with a sample, the more cloudy a sample is, the more cells there is, the more growth there is. Okay, so what we're going to do is we are going to use the spectrophotometer. What it's going to do is it's going to use a beam of light to measure the optical density. So what's going to happen here? So we have our light source in our spectrophotometer. We are going to turn it on and it's going to shine. And that light is going to be collected by a lens. So that lens is going to focus it to a monochromator. This is going to be like a prism or a grate that's going to separate the colors out. We're going to have a wavelength selector, so we're going to select a certain color of light depending on your sample. In this example that we're going to use the yellow color of light. Okay, so we're going to select a certain color with a slit. So this is going to shine through while the rest are not. So we are going to know how bright, we're going to know how much of this light is going into the sample when we shine it through. Okay, so we're going to shine it. If it's a really cloudy sample, it's going to block the light. The light's not going to make it to the other side of the sample. So what happens is the light that's not absorbed by the sample is going to go through and it's going to hit this detector. Okay, since we know how much we started with initially, we know by this detector how much we end up through. So we can figure out how much was absorbed, and then we can use that to make a count of how many cells are in that sample. So this is a relatively quick method, and it doesn't rely on growth. We don't have to wait for things to incubate. Okay, so it's the more cloudy a sample is, the less light, the more cells. It's not as accurate as doing the spread plate technique. We aren't going to get a close number. Also, because it's measuring optical density, it cannot tell the difference between viable cells or live cells from dead cells. Okay, so where the spread plate technique is going to allow us to count how many viable cells are in the sample, this is just going to measure both living and non-living cells, and we can't tell the difference between the two. Okay, so how do we know how many are living versus dead? Well, we're going to use fluorescent microscopy. Okay, we are going to use that specific antibody. Okay, and we're going to stain living one color and the dead another. We've seen this a few times. So green means alive. Okay, red means dead. Okay, so we could get a direct method of counting by literally looking at what's underneath the microscope. Okay, we could literally sit here and we could count each red one and each green one. It would suck, but we could do it. Actually, now we have computers and computer programs that we can hook up to our microscope, so it can see this and the computer will do the counting for us. But this would be a way to directly know how many living and dead cells we have in a sample, which is pretty cool. Okay, that is chapter 6.1. This is a pretty heavy chapter. If you have any questions or if you're confused about anything, come and see me. This is one of the things, because we are doing it in the lab, we need to be able to understand it and do it. Okay, so make sure to come and see me. And that's it for 6.1, and we'll see you in 6.2.